Good morning, everybody. My name is Supanat. I'm a postdoc of the team, and I'm so happy to be here to share my work with all of you. In our lab, we are studying the evolution of several kinds of proteins, and today I will talk about the evolutionary history of a group of arthropod cytoskeleton proteins that have long been debated. Okay, if you look inside an animal cell, you will see this kind of protein network inside. This network structure are called cytoskeleton, and there are three major types of them that are microfilament, microtubule, and intermediate filament. And for our current project, we are focusing on this third group of cytoskeleton component, intermediate filament. We call them intermediate since their size somewhere between those of microfilament and microtubule. And these intermediate filaments are known to play a role in giving cell physical strength or supporting cell movement, something like that. And there are two major groups of intermediate filaments inside a cell, and these are classified based on their subcellular localization inside a cell. That are the nuclear localized lamin, and the other thing that stays inside cytoplasm called cytoplasmic intermediate filament. And although the overall organization and the function of lamin and cIF are different, the protein unit that form these filaments are actually quite similar. And these proteins are grouped within the same protein family called intermediate filament proteins family. And these proteins share the same basic domain architecture, which include a central coil lot domain flanked by low complexity head domain and low complexity tail domain on each side like this. And since lamin need to get inside a nucleus in order to function properly, its tail domain usually include a nuclear localization signal and also this CAF motif that is required for interacting with the nuclear membrane. While in the case of CAF, their tail domain are usually quite simple. Regarding the evolutionary history of this protein, now most people believe that the CF might have emerged from the lamin ancestor by a single gene duplication event in the common ancestor of all Bilaterian. But interestingly, in a group of animals called pan-arthropod, which includes something like insect, spider, or velvet worm, so far no one have ever found something that looks like intermediate filament in most of these animals. So now most people believe that these animals might have lost their cIF since the very early period of their evolution. However, this idea has been made based on the study in just a few pan-arthropod models. And since we know that cIF seem to have some important function in the cells in the other animals, it's quite hard to imagine how pan-arthropod could suddenly lose this protein. So here we decided to revisit this cIF, this pan-arthropod cIF issue again to see whether these animals actually have lost their cIF or not. And since now we are living in the genomic era with many transcriptome and genomic data in our hand, I think it's a perfect time to re-answer this question again. So first we start by gathering available transcriptome and annotated genome from 3,800 species of ecdysozoan, and this includes about 3,700 species of pan-arthropod. And we think this number should be reasonable enough to answer our question. So with this data set in our hand, first we try to pick up everything that looks like intermediate filament based on the overall sequence similarity with our reference IF protein database. And with this first screening, we could retrieve about 1 million hits That is quite a massive number. So we definitely need another round of screening. Since we know that all intermediate filament proteins have a central lot domain, for the next step, we try to pick up only the thing with a central lot domain. And here we use the reference lot domain from this PFAM database and use Hammer to do the job. And after this second round of screening, we could narrow down our candidate to just about 13,000. That is much better. So in theory, what we have in our hand right now should be both lamin and cIF mixing together. So for our next task, we try to separate cIF from lamin. And here we use the machine learning-based tool called Deep Loop to predict the nucleus, to predict the subcellular localization of our candidate proteins. And with this process, we could get this 4,000 hits that potentially cIF in pan-arthropod. And next, we try to clean our candidate list a little bit by trying to remove the sequence with two short coil structure, since they are likely to be incomplete protein fragments. So with all of this bioinformatic screening, we could get eventually get this about 3,600 putative cIF that also include the candidate from pan-arthropod. So now let's have a look at these putative cIF and see where they come from. From the three major groups of pan-arthropod, we only found putative cIF in the group of arthropod and tardigrade, not in onychophorans. And in the group of arthropod, we found that actually most of the arthropod species do not seem to have any of these cIF, and only about 13% of them might have one of our putative cIF. So our result here partially seem to support the previous view of the loss of cIF among pan-arthropod species, and also point out that the species that potentially have cIF might be the minority of the group. So now let's have a look at these minorities and see what exactly they are. From this pie chart, you will see that the arthropod species that potentially have cIF, about half of them are arachnid, while about 1/3 of them are insect. But we also found like a small number of species from different arthropod groups across the arthropod phylogeny. So this is quite a strange but interesting pattern. And this might suggest that cIF might have evolved independently multiple times in many arthropod groups, or it might have been lost multiple times independently in different lineages. This is what the result suggests. So next, in order to know more about the evolutionary history of our putative cIF, next we try to check the phylogenetic affiliation of these proteins. And according to what we know right now, there are two possible phylogenetic groups that our putative cIF might belong. For the first case, it is likely that after the loss of cIF, some pan-arthropod lineage might again re-evolve their cIF back from lamin. And if this is the case, we should expect that our putative cIF should be more closely related to lamin than the cIF of the other bilaterian. On the other hand, if the presence of the cIF is the result of the retention of the ancestor gene, in that case, we should find our putative cIF cluster within the same crate as those cIF from the other bilaterian, and this is our expectation. So for our work, first we try to group our putative cIF based on the sequence similarity to reduce our computational work. And from this reduced number, we pick up each individual candidate one by one and perform phylogenetic analysis separately. So at the end of this analysis, we would get like 1,403 different tree with include each individual candidate inside. And this is one of the example of the tree that we make. It's a little bit hard to see, but in this tree, our putative cIF is clustered within the lamin crate. So in this case, its affiliation should be assigned as lamin. So now let's see the whole result. From this result, we found that most of our putative cIF are actually clustered within the lamin crate. And this result suggests that the cIF in parent arthropod might be something that have re-evolved from lamin, and they are probably different from the cIF in the other bilaterian. But surprisingly, we also found another group of cIF in our putative cIF that are more phylogenetically related to the canonical cIF of the other bilaterian. And this is quite an unexpected result. And this result seems to contradict with our previous view of the evolutionary history of this protein and might suggest an alternative explanation. So now let me put everything together in this slide. According to several previous leads so far, now we know that most bilaterian cIF seem to have evolved from the duplicated lamin, since the duplication event in the common ancestor of all bilaterian. And after this event, both lamin and cIF have been retained in most of the bilaterian lineage. In pan-arthropod, however, we know that the canonical cIF seem to have been lost for some unknown reason. But unlike most previous suggestions, our result point out that this loss of the canonical cIF might not only just happen once, but seem to occur multiple times in many different lineages. But at the same time, there also seem to be some of the other lineage that might retain some of this canonical cIF. And then in the lineage that have lost the canonical cIF, some of the members might again re-evolve their cIF back from lamin. So in overall, our result point out that different groups of arthropods might have different versions of cIF with distinct evolutionary history. Some of them might have the canonical cIF like the other bilaterian, while the other of them might re-evolve their own version of cIF back again from lamin. And this is what we found. But unfortunately, there are still a lot of data that we are actively analyzing in our lab right now. And this is just a part of the result that we could get before this conference. So I still cannot tell you like the complete story right now. And in the very near future, we also planning to examine our putative cIF in real animal as well. And this is one of the examples of the project that we are working right now in our lab. We found that in most Daphnia species, there is only a single copy of lamin. But in this Daphnia purex, we found that its lamin have become duplicated, and one of its copies seems to have lost the nuclear localization signal. So we think this might be an example of the recent evolution of cIF from lamin in a single animal lineage. And I think it would be interesting to examine this animal and see what could be the function of this protein, why the other animal in the same genus don't need this protein, why this animal needs to have this protein. That could be interesting. Okay, before ending this presentation, I would like to say thank you to my buddy, Richard, who helped me carry out a lot of this bioinformatic work, and also my PI, Cassandra, as well as the other lab members who always support me. Okay, that's all we have for today. Thank you for your attention.
Thank you very much. We have time for questions.
Thank you. Nice work and nice presentation. I guess part of your results. I mean, but what's critical here is whether you need both lamin and IF or not. Because it's surprising that you would need a secondary IF to evolve, but then that you've got one species that seems to do without. So do we know that it's essential to have both? And in the secondary one, where does it come from? Does it come from lamin or something else?
So you mean this species, right?
Well, if you go back to your previous slide, kind of your model here, lineage 2 seems to have evolved a secondary cIF.
Yeah.
So there seems to be some pressure to have both lamin and cIF.
Yes.
But then your lineage 3 didn't do that. You don't have a canonical one, you don't have anything else. And I suppose a lot of your question is whether you need both or you can just do with lamin, which your lineage 3 seems to suggest.
That actually might be the next step of our project. Actually, currently we also don't know what is the pressure that caused several loss of CF among arthropod species. And that could be a very interesting question, why some species of arthropod don't have a CF, why some of them have a CAF. But currently, to be honest, I don't know about the selective pressure that make this kind of weird pattern. Some people suggest that because arthropods have evolved the exoskeleton, and we know that CF seem to play a role in giving cell physical strength. So since arthropods already have something to protect itself, they might don't need to rely on CF anymore to make them strength. But so far we don't have any evidence, experimental evidence to support that idea. So for now, to be safe, I actually don't know about the selective pressure.
I mean, we can talk more offline, but I think that's an interesting question, whether that's required at all or not.
Do you think that it's possible that they have gained another protein that was doing this role and that's why it was lost?
Yes. You mean, is it possible that CF
was replaced before it was lost by another intermediate filament?
It's possible, actually, it's possible. I know a case in Drosophila. In Drosophila, there is a group who suggests that the role of CF might be replaced by the protein in the group of topomyosin. Topomyosin might evolve to have quite similar function in arthropod. But again, this experiment has been studied only in Drosophila, and we almost know nothing about the role of topomyosin in the other arthropod species. So I think it would be interesting to examine the other protein that might have some overlapping role with CAF.
Okay, thank you very much.